All right, thank you. Good morning, good afternoon, everyone, depending on where you are. I want to thank all the attendees for being here to hear the presentation, and I want to thank the organizers for inviting me today. I am Shayla West Barnett. Today I'm going to be speaking about generally recognized as safe or GRAS notices for ingredients made by genetically engineered microbes. I am one of two speakers from the United States Food and Drug Administration. My colleague is going to focus on animal food ingredients, and in my talk, I am going to focus on human food ingredients. Here is a snapshot of what I'm going to talk about. I'll talk about the part of the FDA that regulates the safety of the food supply, and I'll talk about what it is that we do specifically. I'm then going to talk about the Federal Food, Drug, and Cosmetic Act. I'll talk about how the act defines generally recognized as safe or GRAS substance uses. And then I'll also talk about what is required under the law in order for the use of a substance in food to be GRAS. I'm then going to discuss FDA's evaluations of GRAS notices that describe uses of ingredients made from GE microbes. And I'll also I'll talk about the safety considerations, how we evaluate those GRAS notices, and I'll walk through a few examples. And then at the end, I'm going to wrap up and I'll point you toward where you can find additional information. So over the past 2 years of the pandemic, most people have heard about the parts of FDA that are responsible for evaluating and approving vaccines and drugs. So that's the drug side or the medical product side of the FDA. I work for the food side of the FDA. I work for FDA's Center for Food Safety and Applied Nutrition, or CFSAN, and within CFSAN is the Office of Food Additive Safety, or OFAS. OFAS administers pre-market regulatory programs that ensure the safety of substances that are either used in foods or on foods. OFAS also administers the Biotechnology Consultation Program, and that program is responsible for evaluating the safety of new crop or new plant varieties. Within the Office of Food Additive Safety is the Division of Food Ingredients, or DFI. DFI administers the GRAS Notification Program, the Food Additive Petition Program, the Color Additive Petition Program, as well as allergen labeling exemption programs. There's a notification program and a petition program. So we have many different programs, all administered by one division, and all of those programs have the same purpose, and that is to ensure the safety of substances that are added to the food supply in the United States. The reason why we are responsible for regulating the safety of the food supply is because of the Federal Food, Drug, and Cosmetic Act, and I'll hereafter refer to this act as the act or the law. When Congress passed the law, they conferred to FDA the responsibility to regulate the safety of the food supply. Not only did the law give us the authority to regulate the safety of foods, it also provided a regulatory framework that provided clear definitions for what a food additive use is and for what a GRAS substance use is. And I'll talk more about that here. Under the law, any substance that is intentionally added to food is either a food additive or a GRAS substance. Under the law, the use of a food additive and the use of a GRAS substance must be safe. And that's an important point, so I'm going to say it one more time. Under the law, when you're using a food additive or a GRAS substance in food, the law requires that that use be safe. The law also requires that for substances that are used as GRAS substances, that in addition to being safe, they meet 2 additional criteria, and that's shown here on the left. In order for the use of a substance to be GRAS, in addition to it being safe, the data demonstrating the safety of that use must be generally available. This means that those data have to be accessible to the public, either in the form of an article in a peer-reviewed journal, inclusion in a textbook, or inclusion in some other type of scientific document or report. In addition to being generally available, the data demonstrating the safety of a GRAS use must also be generally accepted. That means that those data, the safety data, have to be in the public domain, and there has to be evidence that qualified scientific experts are aware of the data and agree with the data. And so I'll summarize that again, that just like a food additive, a GRAS use must be safe. That's required by law. And the law also requires that the data that support the safety of a GRAS use be publicly available and generally available and generally accepted. And what this means for manufacturers is that the information that they rely on to demonstrate the safety of their GRAS substance cannot be confidential. They cannot be otherwise privately held. They have to be out there in the public domain for all to see. I also want to add here that in the absence of a food additive regulation issued by FDA, the law requires that a manufacturer make a GRAS conclusion. So if there's no food additive regulation, in order to legally market a substance for use in food, a manufacturer is required by law to make a GRAS conclusion. Once a manufacturer makes a GRAS conclusion, which is required by law, the manufacturer can then choose to participate in FDA's voluntary GRAS notification program. And that's what I'll talk about now. So we are about 1,000 GRAS notices in. We're at about 1,020. So we have a lot of experience evaluating GRAS notices. And in that experience is included our evaluations of GRAS notices describing ingredients that are made by GE microbes. And so what I'm going to talk about now are the types of safety data and information that we evaluate. So when we evaluate a GRAS notice for a substance that is made using GE microbes, essentially what we're evaluating is the product of a fermentation process. And so I figured I'd start this part of my talk by giving a really quick overview of the fermentation process. In the fermentation process, a production organism that makes the desired ingredient is cultured in a fermentation vessel, similar or maybe different from the one that's shown on this slide. During that process, the production organism makes the desired ingredient. After the fermentation process is complete, the desired ingredient is recovered from the fermentation vessel, and it is then purified. And then that purified final ingredient is used in foods that the manufacturer intends to use that substance in. When we evaluate a GRAS notice for an ingredient produced using GE microbes, essentially what we're doing is we're looking at safety aspects of pretty much every step of the fermentation process. And so that's what I'm going to talk about now. The fermentation process for an ingredient made by a GE microbe starts first and foremost with the identity and the safety of the production organism. When we evaluate a GRAS notice for an ingredient produced by a GE microbe, it is essential that a manufacturer thoroughly and completely describe what the production organism is. Typically, what we see are production organisms that are derived from predecessor organisms that are known to be safe. They have a safe strain lineage. That's not required, but it is something that we see quite a bit. It is essential that a production organism used to make a food ingredient be non-pathogenic. And what that means is that there be evidence that that production organism is not known to spread and cause disease in the human body. It's also essential that that production organism be non-toxic or non-toxigenic, meaning that it is known that that organism, microorganism, does not produce substances that can cause harm in the body. It's also important to know that a production organism does not make substances that can interfere with clinically important antibiotics. Most manufacturers who submit GRAS notices to our program will demonstrate the safety of the production strain and the identity of the production strain by providing a narrative that discusses what's known about the genome of the production microorganism. So for example, they'll provide a narrative that talks about the fact that they know where the organism came from, how it was developed. They know that there's nothing in the genome of that production microbe that is known to contribute to disease or to toxicity or to antibiotic resistance. It's strongly recommended, but not required, that a production microbe either itself be deposited in a well-known repository or be developed using a microbe that's been deposited in a well-known repository. The reason that this is strongly recommended is because repositories are known for thoroughly identifying and characterizing the microbes that they have. And so that's one way that some manufacturers demonstrate that they're clear about the safety and the identity of their production microbe. In many cases, when we evaluate these GRAS notices, the production microbes are modified in some way in order to produce high levels of a desired ingredient. And so that's what I'm going to talk about here. When a production microbe has been modified to produce a desired ingredient, it's very important in a GRAS notice for a manufacturer to clearly discuss how that microbe has been modified, what genetic sequences have been introduced into that production microbe to make it produce that ingredient. What we want to know is where those inserted genetic sequences are from, what organisms are they from, and what's known about the safety of those organisms. We also want to know if there's any extra DNA that has come along for the ride with the inserted genetic sequences that are introduced into the production strain. We want to know what traits, what functionalities are being introduced into the production strain when those genetic sequences are inserted. And something that we want to know is when there are genetic sequences inserted into a production strain, we want to know whether or not those inserted genetic sequences lead to the production of proteins. And one reason why we want to know that is because there are some proteins that either are allergens or have similarity to allergenic proteins. We also want to know when there is inserted genetic information that that genetic sequence is stable in the genome of the production organism, and that there's not the likelihood that that inserted genetic sequence can be transmitted to some other sort of unrelated microbe. And so all this to say, it's very important for the inserted genetic information, the inserted genetic sequences, to be thoroughly characterized and identified. And most manufacturers, when they support GRAS notices, will again provide a narrative that discusses what's being inserted into the genome of the production strain, what functionalities are being introduced, and a discussion that confirms that the introduction of those sequences does not raise any issue or concerns with respect to safety. We also examine aspects of the fermentation process itself. One thing that we ask manufacturers to be clear about is whether or not there are any components of the fermentation medium that either are major food allergens or are derived from major food allergens. And the reason why it's important for us to know this is because we want to understand whether or not the final desired ingredient is likely to contain major food allergens, which would be a safety issue, and would also raise issues with respect to how that ingredient needs to be labeled. Let me also go back. We also want to know about the fermentation process. We want to know how the fermentation process is controlled. What conditions are controlled? What are the checkpoints that are examined for potential contamination? And if or when contamination is detected, what steps are taken to mitigate the likelihood that that contamination will somehow make it into the final ingredient and then later into the food supply? We also need manufacturers to tell us about the composition of the final ingredient and the purity of the final ingredient. This includes measuring the presence or absence or levels of any potential impurities, telling us about any contaminants that may be present. And we also want to know whether or not the production microbe is present in the final ingredient or whether it is absent in the final ingredient. We also expect manufacturers to tell us what foods they intend to use that final ingredient in and at what levels they expect to use their ingredient in those final foods. And then with that information, we expect manufacturers to provide an exposure estimate to tell us how much of that ingredient they expect the human population to consume. And then that exposure estimate is to be compared to well-known safety data and information to substantiate whether or not that use of that ingredient is safe. And so that's a general snapshot of what's included in a GRAS notice. I didn't go down into too much detail. What I'm going to do now is just walk through a few examples of GRAS notices that FDA has evaluated that describe uses of ingredients made by GE microbes.
Hey, Shayla, just
Yes.
wanted to let you know we're getting close to time.
Okay, so I'll just run through the examples very quickly. We've seen a lot of enzymes. We've seen about 100 GRAS notices for enzymes. Same considerations as before. An additional consideration is because an enzyme is a protein, we want to know that the enzyme protein is not similar to proteins that are known to be allergens. We've seen a lot of algal oils. I heard that mentioned earlier in the workshop. We have seen many oils that are made by microalgae, and these oils are made and introduced into foods to provide some sort of nutritional resource, such as docosahexaenoic acid or oleic acid. We're very interested in this case in how those oils are cleaned up after the fermentation process and what the components of the oils are. And we've also seen yeast. We've seen yeast that have been modified by introducing genes from basil and mint plants to introduce sequences that will result in the production of substances that give beer its flavor. I'll wrap up here by saying that the Division of Food Ingredients ensures that food ingredients are safe for their intended uses. That is first and foremost what we exist for and what we do every day. I'll also let you know that our GRAS notification program is transparent. I know I gave some really general examples, but if you're interested in seeing the GRAS notices that we've evaluated, as well as our responses to those GRAS notices, you can do a search or you can go to FDA's website and find our GRAS notification, our GRAS notice inventory. And there you'll find a searchable database where you can find the GRAS notices we've evaluated and how we've responded. And I'll end by saying that if you're a manufacturer that's interested in submitting a GRAS notice, we highly, highly recommend that you request a pre-submission meeting with us. This gives us an opportunity to provide advice and recommendations. It gives you as a manufacturer the chance to give us an overview of your GRAS notice, and we can talk about any concerns that may be apparent from that discussion. If you're interested in requesting a pre-submission meeting, you can request one at this email address shown here. It's pre-market, and pre-market is without the E, premarket@fda.hhs.gov. And with that, I will thank you and I will hand things over to my colleague.
So thank you for the opportunity that you guys are giving CVM to talk about how we regulate genetically engineered organisms for use in animal food. So the first thing I would like to talk about is that in the United States, a substance that is added into animal food can be regulated as a drug or as a food. Drugs are articles included in the diet to treat, cure, mitigate, or prevent disease, affect the structure or function of the animal, and improve animal productivity. Now, food, as defined by the court, is any article included in the diet to provide nutritive value, taste, or aroma, affect the characteristics of the food, or directly or indirectly become a component of food. So one of the most simple examples I can give you today is how a bottle of water can be regulated as a drug or as a food. So it's all dependent on the intended use and what the firm is claiming that the substance is going to do in food. So if the firm claims that that bottle of water will prevent dehydration in animals, we would regulate that bottle of water as a drug. However, the same substance, if the firm intended to market the product as a source of water and essential nutrient in the diet of animals, we would regulate that product as a food. So genetically engineered microbes can be used to produce several different substances for use in animal food, and here are some examples of those. However, the genetically engineered microbes themselves are rarely the focus of a regulatory submission. The substances produced by genetically engineered microbes are often the focus of the regulatory submission provided by the firm. However, the agency is interested in ensuring, like Sheila said, that substances added to animal food are safe and effective for the intended use. So microbial safety and genetically engineered processes are different sections that a firm needs to consider and address in a regulatory submission for use in animal food. There are currently 3 regulatory pathways in which a firm can submit a regulatory package for CVM's evaluation of the substance, and they are the food additives petition process, the general recognized as safe process, in which is the firm's conclusion that the use of the substance is safe, and also the new ingredient definition process, which is supported by the Association of American Feed Control Officials, which publishes an official publication that contains these ingredient definitions proposed by CVM. So today I'd like to discuss some general considerations and questions that the agency would ask firms, and the firm should be able to address when submitting regulatory packages produced by genetically engineered organisms. So like Sheila said, the first question that we want firms to address is the identity of the source organism. The focus of this section in the microbial safety is to evaluate the safety of that source organism. So it's fundamental that we would know which organism is being used to produce the substance in question. So there are several ways that a firm can address the identity of the organism, and they should provide data to substantiate the identity and taxonomical classification that is included in the regulatory package. We also want to know how was that organism selected? Was it purchased from a culture bank, or was it isolated from the environment or from an animal? So we want to know any background that the firm may have concerning the source organism. Part of the microbial safety review also involves looking at the scientific literature and reporting information associated with the safety of the source organism. These safety assessments should focus on the genus species of the source organism and not necessarily on the strain that is used. Food in the United States is not proprietary, differently from drugs, and the safety of genetically engineered organisms are evaluated and regulated on a genus species level. So any background information also on the use of that genus species for the production of substances to be included in animal food is relevant to this section. Also in the safety assessment, the agency is also evaluating potential for pathogenicity and virulence of the source organism. Is the organism associated with any animal or human diseases? Does the organism produce any toxins or undesirable substances? And one important aspect of that safety assessment is to keep in mind that the absence of safety information in the scientific literature does not necessarily translate to the fact that the source organism does not raise safety concerns. It would just mean that the safety information has not yet been published or evaluated. Another question the agency will ask firms is if the source organism is viable in the final market formulation. The agency standard is that the firm claims that no viable organism will be present in the final market formulation, that it would really mean that there are 0 viable source organisms in the final market formulation. We note that some firms propose log reductions as a means to address absence of the organism in the final market formulation, but that is not acceptable. The firm should also provide detailed description of field steps and how they ensure that the final market, there is no viable organism in the final market formulation. Firms usually strive for not having viable source organisms in the final market formulation because food in the United States is not proprietary. So if there's a viable organism in the final market formulation, a competitor farm could isolate and use that organism to produce the same substance. However, if farms decide to have viable organisms in the final market formulation, the agency will require the substance produced by genetically engineered organisms to come in as a food additive petition, and consequently, the safety evaluation will be conducted within that FAP. The second section that is important in the evaluation of substances produced by genetically engineered organisms addresses the safe use of these substances for use in animal food, and it's the molecular biology review. This section will focus on the engineering process itself, so the farm should describe in detail how the source organism was engineered to produce the substance that will eventually be included in animal food. So part of that description involves detailed information on the genetic material that was introduced and what is the source of that genetic material. Is it synthetic? Or is it from a gene donor? And in that case, are there any safety considerations that should be addressed? The farm should also disclaim any intentional changes to the nucleotide sequence incorporated into the genome and the source organism. Farms should also be able to describe the technology that was used to develop the source organism and the techniques used to introduce the plasmid and select for potential source organism strains. We would also like to know the firm should describe the analytical method used in the bioinformatics approach to characterize the DNA insertions, deletions, and edits, and the sequence of nucleotides that were inserted, deleted, or edited in the source organism's genome. An important point to be addressed is if there are any metabolic changes to the source organism that will be relevant. And an example for that is the production, many cases we see in the production of biomasses that are intended to be used as a source of amino acids in animal food. They involve metabolic changes in the organism to accumulate specific amino acids. In those cases, the question is always, are there any unwanted toxic or undesirable substances that are also accumulated due to the change in the metabolism of these organisms? Regardless of what substance is evaluated for use in animal food, whether produced by microbes or not, part of the regulatory evaluation for the safe use of a substance is to obtain a detailed description of the manufacturing process to produce that substance. The agency should be able to understand step by step all manufacturing processes and procedures used to produce that substance for use in animal food. So for substance produced by genetically engineered organisms, part of the manufacturing process should address what is the composition of the fermentation media. In the development of fermentation products, considerations should be made to ensure that all substances included in the fermenter are acceptable for animal food use. The farm should list all these substances in the regulatory submission and address their regulatory status by providing a certificate of analysis for each one of these substances. So in general, bioengineered organisms and the substances they produce are evaluated by the agency on a case-by-case basis for use in animal food. So if you have any questions, CVMs would like to encourage farms to come talk to us and discuss the specific issues in the beginning of the developing a product rather than at the end of the process. So if you have any questions, you can email us and schedule a meeting so we can have discussions that are pertinent concerning specific products in genetic engineering processes that you are developing. Thank you very much.
Great. Thank you so much, Diego and Shayla. So now we have time for some questions. If you'd like to both come back on screen. And thanks to everyone who are putting their questions in. We'll do our best to get to them. I'd like to start with one for you, Shayla. Although, of course, Diego, if you have some perspectives on this, you're welcome to add to it. Can the extensive GRAS requirements for GE microbial ingredient production be contrasted with what is typically required for non-GE derived food ingredients. And for example, there are a lot of allergenic non-GE products on the market. Are they subjected to similar scrutiny?
So if I understand the question correctly, what I'm being asked is whether or not ingredients that are produced using non-GM means are subjected to the same sort of evaluation as ingredients that are produced using GM means. Do I have the question correct?
That's how I'm reading it as well.
Okay, the answer is yes. So I think in our program, we don't see ingredients that are produced by genetic engineering as inherently any less safe than an ingredient that is produced any other way. The only thing that changes is essentially what it is that we would need to see or have access to in terms of information to demonstrate safety. For a non-GE produced ingredient, we still want to understand the identity of the ingredient. We want to know how it was manufactured, what the final composition of the ingredient is, and how much of it is used, how much people are going to consume. For an ingredient from GE production, we still want to understand those same things. The only difference there is that those genetic engineering techniques are part of how that ingredient is made. And so that's why we want to understand where the genetic sequences come from, what the genetic sequences are, and what they do in the production organism. But on the whole, I don't think that our program regards those ingredients as any different or one any more safe or less safe than the other.
Great, thank you. And then this is sort of a overall, regulatory philosophy question for both of you that I'm sure we've all discussed a lot. Much of the world uses a process over product approach. Is this in disharmony with the regulatory framework that a lot of the US uses, which has a product over process approach? And, how can we, mitigate the differences between those approaches?
I'm not familiar with what the process over product approach is. I'm sorry.
Oh, no worries at all. We can always talk about that later when we have more general discussion, but generally it's just as you described, how your office considers both products made from GE and non-GE microorganisms the same. So that would be a product-based approach.
Okay.
So you're not starting out the regulatory process by looking at things because they're genetically engineered.
Okay. Yeah, I think then my answer would be the same as the answer to the previous question, where really what matters to us is the ingredient itself. And of course, we pay attention to process because how something is made influences what the identity of that final ingredient is. And so that's where our wanting to understand how something is made comes from. In order for us to understand what an ingredient is and be able to assess or agree with a safety conclusion, we need to know how it's made. And so I think that's the answer to that question. Diego, do you have a different take?
No, we take the same approach. We are focused on the product itself because we want to really know how that substance is being produced to adequately evaluate any safety concerns that can be associated at the final market formulation.
Thank you. I am wondering if you can share if there are any examples of genetically engineered microorganisms that are themselves determined to be GRAS for human food ingredients, which there was a link posted in the chat for the database. But then further, if you could elaborate a little bit on what would be needed to have a GRAS notice for such a strain.
Sure. So I am not aware that we, through our program, have evaluated a genetically engineered microbe for use in food where the genetically engineered microbe is the ingredient. In short, what would be needed to demonstrate that use of that genetically modified microbe is GRAS would be the same thing that would be needed for the microbes that we've evaluated that are used in food that aren't genetically engineered. So we see a lot of the so-called probiotics, those healthy, beneficial microbes that people add to foods. If we were to see something like that were to be genetically engineered, the safety considerations would be the same. We would still want to know the identity of the microbe, including how it was modified. We would want to know that microbe is non-pathogenic. non-toxigenic, not of any concern for antibiotic resistance. So really the same safety considerations with the added consideration that if it's genetically modified, we want to know how it's modified in order to understand the identity and then be able to assess the safety of that ingredient.
Thank you. And then Diego, I'm wondering if you could answer the same for animal feed. Are there examples of where the microorganism itself is used in animal feed or has been approved for use, and what would be needed to gain that approval?
Like I said in the beginning of my presentation, it's rare that the organism itself would be the focus of the regulatory submission. It's usually the substance that the organism produces that is focus of the submission. So I can think of an example that the organism is what's being regulated. We have 3 different regulatory pathways to address safe use of substance in animal food. So they differently a little bit from CISTIN, the whole gamut of the things that get submitted to us come via GRAS, but the things that do come to us via GRAS, they are approved or acceptable for use in animal food for an intended use. So the organism itself wouldn't be GRAS, or the substance itself wouldn't be GRAS. It would be the use, the stated use of that substance at a specific inclusion rate that would be GRAS. So it would be a little bit different and like I said, microbes are usually regulated as part of the manufacturing process of regulatory submission, and they are not usually the focus of the submission itself. Does that answer your question?
I hope so. We're going by, what the attendees are asking. Let's see. I think we have time for just 1 or 2 more questions. We've got a couple of questions that are kind of asking along the same lines on what happens when there isn't information that's available. So we have a lot of cases where there's an existing product and or an existing microorganism that we already know about, and there's new products that are similar. And so we have information available. But what happens when there's truly a novel organism? And the example one of the attendees provided was a novel edible algae, for example. What types of safety data might you ask for, or what kind of regulatory process might you be looking at for those truly novel products?
For a truly novel product, I think the important thing to point to is the fact that whether something is novel or whether it's something that maybe has been used for a while, in order for the use of that ingredient to be GRAS, it still has to meet all of those criteria. It still has to be safe, and the safety data that are relied on for the use of that ingredient have to be out there in the public domain, and the scientific community has to agree with those safety data. And so even if something is novel, if a manufacturer can make the case that there's enough data and information out there in the public domain that the scientific community knows about and agrees about, and that supports the safety of that use of that ingredient, then we're good to go. However, if there's a situation where there's not enough publicly available safety data, that use of that ingredient cannot be GRAS. The manufacturer in that case, if they can still demonstrate safety, but it's just a matter of the information not being publicly available and agreed upon by the scientific community, the manufacturer in that case might want to go the food additive petition route. And that route requires that they submit a food additive petition and FDA evaluate that petition and issue a regulation prescribing safe conditions of use. So regardless of whether something is novel or not, the requirements for GRAS are always the same. And that's something that a manufacturer has to consider when they're choosing what route to go through when they come to FDA.
I'd like Diego to respond to that as well, but for a clarification, if a product goes through that novel additive route, can following similar products then go through the GRAS route? Or do they all have to then go through the...
Me or
no, sorry, that's for Shayla, yeah.
Okay.
Sure. So it really depends. If over time, scientific information, safety data become publicly available and are vetted by the scientific community, then those later manufacturers may be able to submit a GRAS notice and may be able to be successful. It really still depends on whether or not they're not are just our data out there, but adequate safety data that make the case for safety and that we know that the scientific community is aware of and agrees with. And so as science changes over time, maybe data becomes available in the public domain that might not have been available earlier on. Maybe that puts the later manufacturers in a position to be able to make GRAS conclusions, but it depends on what scientific data are out there.
Thank you. That's fascinating. Diego, could you speak just a little bit about these more novel products, and then we'll close the session.
Like Shayla said, the requirement for GRAS substances is that the information would be publicly available. So in the case of something that is so novel that there is no information in the public literature or the scientific literature, it will push the substance to be regulated as a food additive. And the information that will be necessary, it really depends on what the data that the firm itself has generated already at that point. So we can have a little bit of more information on what we have to look at. Is it related to an existing species or not? What do we know about sequence homology to those familiar strains or familiar genus species around that realm? And that will really craft what kind of information we want to see from that. So I can't give you a general response that would address, oh, this is a checklist of things that we would look at, because it will really depend on what comes from the firm and what kind of information they have already generated from that organism.
Thank you so much to both of you. And now we'll move on to our next session.